Hi Hires, we are in the Metabolism and Survival unit and we're looking at how we use and improve microbes. So when we talk about microbes, what we first have to realise, and I hope that you have a good handle on, is that they are found everywhere. I mean, it is amazing where you will find a microbe. You can find microbes in subglacier lakes, and you will find them in the Atacama Desert, you will find them in high salt, you will find them, well, they've discovered you can keep them in space. That was the bio-rock stuff that went up with the International Space Station, fascinating stuff. But basically, bottom line is, if you live in different places, you have different chemicals that allow you to do that, and different proteins and different enzymes. So if you have this potential, then you are looking at a huge thing that we can use. Okay, but what we've also got is the next level, which is, can we actually change what we have? And the reality is now we can. Okay, so we talk about the wild type or wild strain of a microbe if it's been taken directly from its natural environment. Okay, so that's the thing which we start with. And then we start messing with it, and that would then become an engineered version. So to start with how we might actually try to change what we've got, the first thing you can do is, it's pretty much a hit and hope version. So you basically just expose the microbe to a whole pile of mutagens, or one mutagen, but what you're trying to do is create mutants. And those mutants will have different alleles, different potentials, and they may be what you want. It could also be something that destroys them, but to be honest, we're not that nice about liking bacteria in the same way that we do for bigger things. Okay, so you can use any of the mutagenic stuff that we've talked about before, so any form of ionizing radiation, any form of chemical mutagens, of which there are many. Okay, the problem is that obviously you don't control what mutations you get. All you're doing is hitting the DNA and hoping that you're going to get something different that you like. And then quite often these strains that you make, these mutant strains, they're not genetically stable. It means that they will revert back to the wild type if you leave them in culture. So it's a good starting point, but it's not really targeted. If we're looking for more control, what we can do is horizontal gene transfer. So that means that we are taking, in the generation that we're looking at, we're going to drop in the DNA that we want, okay? And there's several different ways that the bacteria or microbe can take these in. And the first thing is transformation. So what we do with transformation is basically surround it with a soup of fragments that have come from the cell that you want to go into the new cell. So if they take up these DNA fragments, then some of them can be incorporated into the chromosome. And now when they're in the chromosome, they're part of the inheritance. It's again a system where you are playing the numbers. You'll do a lot of fragments, and what you might have done is just use breaking up another cell to do this. But what you can do is PCR, and so amplify a particular fragment and then hope that that one is the one that's going to be taken in and incorporated in a way that it'll express. Okay, second type of horizontal gene transfer is transduction. So in this case, what you have is a virus acting as a vector for the DNA or for the code, because it can be a retrovirus, which means it is taking it as RNA, converted to DNA, and then back into the code. So this little bit of nucleic acid is injected into the bacteria, and then this can now be taken up and part of the code. Third possible way of doing this is something called conjugation. And conjugation, when this was actually discovered, that how and why this was happening changed a lot of biology thinking in terms of bacteria. So what they discovered is that some bacterial cells contain a plasmid called the F plasmid, the fertility plasmid. And what this plasmid does is allows the first bacteria to create a tube made up of a pilus, which is one of the connections that they use to anchor. Okay, and copy across sections from the plasmid into another bacterial cell. And this is how you get high-speed transmission of things like antibiotic resistance, and it is really important as a system. Okay, so once you've got your microbe that you're looking at, what you can also do is some selective breeding. Now that sounds in some ways a little bit odd, because you go, Well, selective breeding means picking parents, but there are some microbes that can use sexual parts of the cycle as well as asexual. And so yeast goes through a kind of two possible phases of this one. So you can still do selective breeding, but it is not possible in all microbes. So we now have the next level up, which is if we can actually use all of these other techniques and then bring in our recombinant techniques, which means taking DNA, combining it with something that it wouldn't normally be combined with, then it's possible to make something that is improved in exactly the way you want. Not a hit and hope, not a potentially this might work, not a taking its time to work through different generations, but actually saying, if we get all this right, we can do it in a single step. And that's us.